Good morning, everybody. We're going to start. It's a little after 8:30. Tomas, Sam, and I welcome you to our integrated chemical imaging workshop. We have a very packed agenda because it's only one and a half days for this workshop. Quickly, I'm going to go over a little bit of logistic questions. So if you in the audience, if you guys have any questions, please wait for us to give you the microphone, because otherwise the people who are online will not be able to hear you. And then the breaks, please make sure that you come back on time so that we can get going right after. Okay, so today we are going to hear a lot of talks. We'll start off with a lot of capability talks, and then we'll talk about data from how we can get all these different imaging data from different techniques that also we will describe, and then we'll go into data integration. There is a workshop tomorrow, and then we'll wrap up with some future, what is the vision for chemical imaging. And primarily, we are going to discuss few grand challenges: the imaging challenge and the data integration challenge. Tomas is going to go over these challenges today. Something that I want you to keep thinking about during this workshop is, as you hear more and more about these challenges, please try to keep in mind questions that you can ask that will help solve these challenges, and how we can integrate all this data, and what kind of platforms we need to integrate these data. Okay, with that, I'm going to introduce our first speaker. It's Tomas Varga from PNNL.
Is it working? Yeah. Thank you, Arunima. Hi, everyone. Thank you for coming and joining. So I'm Tomas. I'm here in EMSL, and so I will introduce this workshop. I'll talk about why we are doing this chemical imaging workshop, talk about some of the science drivers, research needs that we have for why we are integrating. Then I will talk about some of the key measurement capabilities that can be considered for chemical imaging and integration. And then some of the specific challenges that we have for integrated chemical imaging. Then I will switch gears and kind of want to set the tone for our discussions of the grand challenges that exist for integrated chemical imaging. And then in the end, we have some thoughts about what would be an awesome outcome of this workshop. So why are we doing this now? At this point, we have a great number of well-developed and integratable imaging capabilities, but we still lack integration across all the measurement methods. We also have a persistence of largely manual data processing steps, and a shortage of scaling approaches that is able to bridge the gap between the microscale information and the landscape scale. So so if you want to be able to understand and ingest all the microscale information and process knowledge and inform the modeling, then we need to basically need a good plan to synthesize the data, integrate all these methods, so that we can provide that bridging. So and we need to collaborate. We need collaboration across disciplines, experimentalist modelers, also to develop robust methodologies that can be then allow to develop workflows that are solid enough to be reproducible and can be high throughput and can be automated. So if we want to make all the microstructural information available, accessible to model development, then things like the interoperability of the data, the fusion of the different modalities, data from different measurements, and the scaling of the data will become critical. This workshop is also timely because this year we had a series of in-house chemical imaging visioning meetings that we started discussing these needs. And also in April, we had a ESS PI meeting breakout session with the same topic. So today we are able to use the outcomes of those meetings in our discussions today and tomorrow. And so finally, multimodal imaging is hard. If we have a visionary plan of how we exactly do this, we can have a better chance or impact on giving the scientific community service and leadership. It can also affect or inform our investments in techniques and methods. So in our discussions, we identified the following scientific needs, or we can call them opportunities for chemical imaging. And these we grouped into method drivers, or we can call them imaging needs, as well as data/model drivers. So what do we want out of the integration? We want species resolved as well as spatially resolved information. We want dynamic information, dynamic measurements that give us in situ, in vivo information, and the response of systems to perturbations. So sample environments are important here. We need good bulk data that can validate the microscale and molecular data. We want to identify those microscale processes that control macroscale processes. And I mentioned the robust workflows. We want to develop workflows that can be automated and maybe standardized. And then for measurements, specifically sample holders that make registration between modalities easy or easier are required. From the data and model side, we want to be able to merge, synthesize all that structural, morphological, and chemical information and register those across scales. We want to correlate, parameterize, and scale data for databases and modeling. And we want to do that, making data operable for modeling without human bias. Data fusion is important because we have multiple techniques of the same measurements on the same sample, but also if we think of a high resolution data set on a larger region and collected across the whole region, we need to be able to fuse that into one single high resolution data set on that larger region. Real-time analysis for screening samples. So if you think about wanting to inform your measurement and to find unique features, for example, then kind of this on-the-fly analysis for screening and loop back into the measurements is important. And then finally, we need new quantitative microscale approaches to derive parameters for predictions. I wanted to just stop for a moment and talk about what do we mean by chemical imaging. So for me, chemical imaging is obtaining some sort of structural or morphological information and chemical information on the same sample. This can happen using one measurement modality, one technique. Classic example would be SEM with EDX, or it can happen with using multiple modalities. And so then integrated chemical imaging is the combining two or more measurement modalities or measurement imaging technique and a modeling tool to get more information out from a specific environmental system. So after so many words, I want to use some images and highlight some of the first EMSL capabilities that are can be considered for chemical imaging and integration. This is not a full list, and but EMSL has a range of microscopy techniques, X-ray techniques, mass spec imaging methods, and spectroscopies that basically cover about seven magnitudes of spatial range resolution. And So I also have some headshots, and I'll, for the sake of time, I'm not going to introduce staff, but I mentioned first names, and please reach out to us if you want full contact information of any of the instrument scientists mentioned to get in touch. So for electron microscopy, we have a state-of-the-art suite of SEM and TEM systems. They can all be integrated or already integrated for chemical imaging, SEM and TEM with EDX, or TEM with EELS, electron energy loss spectroscopy, to get chemical information on those systems. SEM more like a millimeter micron range, and the TEM down to atomic scale. And we have a variety of sample environments for in situ measurements. So Libor, Odetta, and Yaobin are our leading scientists on microscopy. And you will hear from Odetta an example of a use of SEM for data integration, as well as Devin will also address that. We have some other microscopy capabilities, such as laser caption, dissection microscopy, that can be paired nicely with omics on the dissected section or piece. Laser scanning, confocal microscopy, fluorescence microscopies. These are led by Will and Dehong. For example, fluorescence microscopy is already chemical imaging. If you use the appropriate fluorescent tag, then you can get quantitative measurements of a protein within a cell, for instance. And we have helium ion microscopy with Shuta, which is not by itself, but if it's paired with a mass spec capability like SIMS, it can be chemical imaging. We have X-ray, CT, computed tomography. I lead this technique, and so it's depending on sample size, it's a great tool to study soil pore structures, as well as root architecture, rhizosphere systems. And we are very interested in pairing this capability with chemical. By itself, it's morphological, density-based, but with chemical modalities, it can be really powerful. Next, we have EMSL has a great suite of mass spectrometry imaging capabilities. Chris, Dushan, Kevin, Greg, Maria, and Rosie are the instrument scientists leading different techniques here. It really covers a range of scales. Dushan later will talk about application of multi-MSI, and Chris will have a talk tomorrow on the visual omics platform for biomolecular characterization. And some of the mass spec based capabilities are atom probe tomography with Denny, which is a three-dimensional atomic scale imaging. TOF SIMS with ZUA, nano SIMS, these are already chemical imaging capabilities as well. TOF SIMS has it's a surface technique with pretty good lateral but very high depth resolution, and nano SIMS with Jeremy, better than 100 nanometer lateral resolution, providing chemical images. So what are some of the challenges of integrating all these techniques? Instruments, a lot of instruments are still largely utilized in a standalone fashion, and not so much as integrated multi-method capabilities. Today we'll hear about, the next slides I'll show some integration efforts, and we'll hear a list of multimodal talks from Odetta, Devin, Swaroop, Jocelyn, that shows successful integration of some of these techniques. We need algorithms to quickly and seamlessly integrate and co-analyze data from these different imaging methods. Sam Webb will talk about the software tool SMAC that can be applied for multi-image data, X-ray, microprobe, microscopy, IR, mass spec imaging. And then also we'll hear from Peter later about using multimodal image data for multimodal predictions. And finally, we need scaling relations that can translate microscale information to landscape scale. And we will have a talk by Angela today after the coffee break about scaling insight from nanoscale imaging to inform landscape scale. So you can learn a lot about these issues already. So I'll give you some specific examples of multimodal integration. This is a slide from Arunima. So Arunima and Jade are responsible for the microenvironments at EMSL, such as the rhizochip, the terraforms, the portal chip. And these are great examples where these reduced complexity systems can be paired with imaging techniques such as multi-MSI or synchrotron-based techniques such as X-ray fluorescence microscopy or XANES. We'll hear specific science about these in the next couple talks. And then, so I mentioned we are really interested in integrating EMSL capabilities. So we are using the high brilliance of the synchrotrons and tunability of the X-rays. In the third talk, Jocelyn will talk about capabilities at SLAC, where we are able to pair EMSL microscopy or mass spec imaging with X-ray absorption spectroscopy and fluorescence imaging at the synchrotron. At the APS Argonne, there are also a suite of capabilities that they offer for the environmental sciences from their Eberlite effort. Unfortunately, Zulf Infrak couldn't do her presentation today because of logistical reasons, but please reach out to me if you would like to learn more information about the different imaging and spectroscopy techniques that are available at the APS. Here's a list, and I can provide these. We are also collaborating with the APS to pair imaging and spectroscopy techniques. So I'll highlight a few examples of the specific challenges that we face in data integration or chemical imaging integration. These slides we use in the roundtable discussion, so I'll just highlight them, can dive into them deeper. One is the fusing 2D and 3D data. So we are interested in correlating CT data that is grayscale, morphological, with those, for example, from SEM EDX that are 2D chemical maps obtained on a reduced number of slices. The goal is to expand the limited 2D chemical information into 3D space and predict the whole chemistry in the 3D object. And so the challenge is here, of course, the method differences. The XCT is fast 3D, medium resolution, single channel. SEM EDX is slower, 2D, high resolution, multi-channel. And in addition, the sample prep involves embedding, fixing and cutting slices from the sample. So we have geometric misalignment, warped cut planes, missing sample pieces. All these need to be accounted for in the computational tool before data fusion. Another example is a correlation of optical microscopy with mass spec imaging. This is from Arunima and Chris. And so some of these challenges are pretty general because finding the same area with a different instrument is a challenge. And co-registering the data between the different resolution images. The MALDI is about 100 micron scale, spatial resolution. Optical microscopy is much higher. And then also the sample distortions to account for. By the way, we'll hear about more about multi-MSI from Dushan using spatial metabolomics. And speaking of metabolomics, we'll also have a talk about multi-scale, metabolomics platform for plant-soil interactions from Malak tomorrow. Another challenge is the multiple scales. So this is an idea that, so this is inspired by this paper here, where you would have the whole root structure imaged and then take subsamples of them for micro-CT. You need to co-register to the whole volume, and then embed samples for micro-CT, create 2D sections to do microscale chemical imaging, and then go down one more scale just on the root for maybe nanosims or laser ablation, IRMS. So the difficulty of registering or correlating the images between the different scales, as well as the multiple steps involved, are definitely a challenge. And then we'll hear from Odetta about this 3D soil system characterization. But here I just want to highlight the challenge and the need for high throughput and automation. If you think about making a sample that you want to maybe stain the organics to stand out in the X-rays, and then embed the samples in resin, do the CT scan, create the thin sections, then generate the high resolution chemical maps in a microscope, it can take a couple of days. So it is something that needs more high throughput. So to sum up the grand challenges that we can discuss is, one is the spatially and species resolved measurements that are critical for understanding the biogeochemical processes. And there is insufficient integration across imaging and spectral methods. So this we can call our imaging challenge. And then on the other side, data processing and integration approaches are largely manual. Data needs to be accessible and interoperable, need more standardized data, and we need scaling approaches that link microscale observations to landscape scale. So this would be our data / modeling challenge. And these are just individual slides of, of course, the imaging challenge, where we want to image individual and coupled biogeochemical and biomolecular processes in soils, rhizosphere, the whole environmental science. Here, these points are already showed on the fourth slide, basically our opportunities or drivers. And from the ESS-PI meeting breakout session, we gathered some feedback from the participants about the needs, what need to be addressed. And so they highlighted scaling, addressing heterogeneity, time and throughput dynamics, and the need for representative and standard data. And you will hear more information from Chris Anderton tomorrow about some of these the outcomes of the breakout session meeting. So one thing that we can discuss today also and tomorrow is which methods are best suited in your research to address these challenges. Then the data modeling challenge. If you want to link the complex molecular and microstructural process observations to macroscopic models, then we need to figure out how to bridge the spatially resolved microscale and molecular data at larger scales. So again, these points are the scientific drivers listed on slide 4. And from the breakout session, the community noted the needs of course, AI, machine learning, interoperability, more software tools, need for data access, and again, need for representative and standard data. We'll have a series of multimodal and automation modeling talks by Sam, Peter, Devin, and also Maruti. So we'll dive more into that topic. So I want to close with this slide. A vision statement like this would be, I think, appropriate. We want to be able to perform integrated chemical and physical imaging of biogeochemical processes and interfaces on the micro to nanoscale and nanoscale in real time to inform modeling from the molecular to landscape scale. So we would like to kind of articulate this vision in this workshop and into our workshop report, hopefully a perspective paper. If we could put out needed approaches, methods for the immediate as well as 10 years out, that would be a great outcome. So we would like to maybe put together a report, which could be like a white paper to a perspective paper, kind of serve as a draft for it, and then hopefully publish a perspective paper. So if we can plan ahead like this, it can really be a great. We all can achieve better impact if we have this strategic plan to serve the scientific community, and that's our hope. And so with that, I'd like to thank the team here at in EMSL who did the chemical imaging visioning meetings: John, Emily, Amir, Swarup, Chris, and Odetta. The ESSPI meeting breakout session was chaired by Chris and Ken Kemner from Argonne. And I'd like to thank the organizers, also John, Odetta, Arunima, Zoe, and Sam. And then for this workshop, the comms team, Linda and Genoa, Heather from Community Affairs, and Stephanie for the financial planning. This workshop couldn't have happened without them. And I'd like to thank for funding from EMSL and BER. Thank you for your attention.
Thank you, Tomas.